Good afternoon, everyone. Thank you for taking the time to join this call. We are excited to start these talks, which are about the HIVE platform and some of the tools and resources that have been developed and how they have been used and can be used. So today's talk is by Dr. Konstantinos Karagiannis, identification of defective viral genomes using NGS data on HIVE platform. This meeting will be recorded and made publicly available. Just a couple of sentences on HIVE. There are two types of portals. The public portal, which we maintain. Anybody can use it. You have to create an account and so on. And also there is an FDA-only portal, which means to access FDA HIVE, you have to be an FDA employee. We have a research collaboration agreement with FDA to co-develop HIVE and work together on the scientific and technical aspects. Contact information is below. Dr. Karagiannis, FDA email address, my email address, and Dr. Jonathan Keeney, who also is the HIVE platform lead at our end. Today, we are going to try something new, which is always interesting and fun. We want you to use Easy Retro. So Easy Retro is a way to interact during the talk. The chat box is disabled on WebEx. All the discussions we want to do through Easy Retro, I hope it works. So the way it works is that you can add new cards here by clicking the plus button here, suggestions, action items, questions. Most likely you will be adding the suggestions box here. You can add new cards here in the suggestion, or if you have any questions as the talk is going. I, in the background, will try to collect all of that, and when Dinos has finished his talk, we will ask him these questions and he can respond. Some of the questions we might be able to respond as the talk continues. With that, we will start. If you have issues connecting to Easy Retro, you can call us or send me an email. Again, the email address is mazumdar@gwu.edu, or you can send also an email to Dr. Jonathan Keeney, and we will try to help you as the talk continues. With that, I'm going to pass on Dr. Karagiannis. He was going to show his screen now, so I'm going to stop sharing my screen here, and he should be able to start sharing.
Hi, Raja. Thank you for the invitation. Allow me a couple of seconds as I'm trying to share my screen here. I don't seem to have the sharing button enabled here. Not sure if I'm doing something wrong. We tested it a few minutes ago. Okay, now I see it.
Okay.
Okay. And I hope you're all seeing my slides right now. So first of all, thank you, Raja, for inviting me, and thank you for organizing this series of seminars. So this seminar is going to be focusing on the defective viral genomes using NGS data on HIVE platform. The HIVE platform here is the center of the talk, of course, but you know, it's only exciting and it's only useful for scientists and for all of us who are trying to answer these biological questions when we look at it from the perspective of the science and the fun that it surrounds it, rather than just the purely engineering and software development aspect. Nonetheless, there's a lot of development performed by both my group and Raja's group for the platform. So we're very excited for this type of seminars. So without further ado, I just wanted to just jump to the next slide. Just a disclaimer here for the FDA that all the views they're my own and don't represent any FDA's views or policies. So I'm required to say that. And having said that, let me just jump to the next slide, which is the first question, I guess, that some of you or many of you might have as to what is a defective viral genome, or what is a DVG as we call it for short. Even before trying to address that question, let me take a step back here and explain that this is a relatively recent term. In the past, these DVGs, as we call them now, they've been characterized as DIPs, or defective interfering particles. But due to the expansion of their roles and the fact that their role was not only interfering, as has been proved through some studies, the term that prevailed right now is the defective viral genomes. And after some recent publications, I think the consensus that is coming, that the field is coming into, is that these are genomes that are practically unable to complete a full replication cycle, and that's why they're defective. And that definition doesn't really give any clues as to whether they're as to what is their function, rather that what's their the outcome of these changes of the genomes. So, frankly, there are three categories. There is a category of DVGs that have been originated through some mutations, either that being 1 or 2 point mutations that somehow affect the replication cycle so drastically, so they result into genomes that cannot complete this cycle, the replication cycle. They can be either that or just hypermutated regions, just regions that are highly mutated. This is something that is often observed in some RNA viruses. And of course, this is a new term that I introduced, RNA viruses, but I hope that we are all aware that these DVGs is a phenomenon that is mainly observed in RNA viruses, mainly due to the low efficiency of the enzyme of the RNA-dependent RNA polymerase. And I was at the point of the hypermutated regions, and the third here that's explained as one of the 3 examples of the mutated type of DVGs is just a simple frame shift, and that's what the red region here represents, just a red frame shift that can lead to a premature stop codon, messing up practically with the full replication cycle of the virus. There's also the deletion version of a DVG, and either deletion or insertion is introducing big gaps to the genome. Deletion either because mainly because of the jump in the polymerase, but this can lead to thousands of base pairs missing from the genome, or insertion, practically jumping back to an earlier point of the replication process and introducing insertion of the same genome to itself. And third version is the copy pack or snapback DVG. It's slightly more complicated. So, and this is mainly what drove the development of the specific tool that we're going to be talking about today, the DVG-profiler. And they're referring to a case where the genome practically, while it's been replicated, the polymerase, it just jumps back to the template that has been created and then continues backwards and creates this stem loop configuration. And you can see here that the representation that's hopefully that's going to be helping all of us to conceptualize this phenomenon a little bit better. So this is a typical copyback DVG, and I will soon explain what is a simple difference between snapback. You can see that it happens when the RDRP, the RNA polymerase, it jumps for some reason from the one point of the the genome, or the antigenome in this case, and then instead of continuing or either at a later point of the antigenome or somewhere else in the same template, it actually jumps back to the template that it's creating itself, and then continues towards the end of it, since now we're in reverse direction, right? And that means also that it creates some complementarity, like this section of the genome is going to be created twice, the reverse complement version of it, and it's going to be able to create this stem loop as we see it here. So practically, and this is the information that we need to define the difference between the copyback and the snapback. Snapback is when this stem loop is practically zero, or this complementary side of the stem here, it's the full extent of the DVG. So if it jumps back to the same place and goes all the way back, this is practically what's called snapback versus the copyback. Minor technical detail, but just a matter of differences in some of the effects that these two have. Not that but in reality, this will not affect the mathematical approach. So there are many ways that these DVGs can be created. These are either the stochastic mistakes of the polymerase that can lead to these point mutations, or the hypermutated regions. There can be reasons like the ones have been observed in the Sunday virus, here explained for the genomic variations, where when a nucleoprotein of this virus has been observed to have a single mutation, seems to generate more of these DVGs. There is certainly correlation there. The mechanism, I think, it's yet to be described fully. There's also a case of some structural variation that affect the generation of these DVGs, like the PPX domain from the lymphocytic choriomeningitis mammarinovirus, where when this domain is present, it just leads to more interfering particles, as they are called in this specific case. They are favored versus the full structural genomic sequence. And finally, the template switching, which also explains the copyback as we were describing it in the previous slide. So there are many mechanisms, but overall, the whole underlying concept is that these differences, they all lead to genomes that cannot fulfill a full replication cycle. And there have multiple effects. As I've already mentioned, they've been originally described as interfering particles because they've been observed to interfere with the full circle of the wild type virus, and this can be done in different ways. Mainly and more intuitively, I would say, it has to do with the fact that it actually competes with replicating resources, right? And the kinetics of full replication cycle of something much shorter, like a DVG, like a copyback, or a genome with a big deletion, that cycle is faster. The kinetics are in favor of these DVGs. So in this way, the DVGs would just exhaust the resources of the wild type virus. And that was a traditional observation. Since then, there have been more and more evidence for other effects of DVGs. Of course, there's evidence that they also trigger some signaling pathways, like pattern recognition receptors, as described here in this publication. And of course, there is also some evidence for persistence, and this is interesting. The last step in the persistence is also observed in HIV, and I have here a slide that includes the figure of one of the publications. Interestingly, this is a publication where Dr. Fauci has co-authored right before the pandemic, practically. This is from early 2020. So he's been working actively on this field, which is interesting that more and more people are looking into DVGs, even on HIV that's been heavily studied. And what they've seen, practically, what they notice is that these DVGs, they do appear in practically most of the stages of the HIV infection, both early and late. And there are of various types. There are single point mutations that called create DVGs, and there are deletions as they are as they're reported here in this figure. And what they observed is that these DVGs actually they are the main reason for some silent pools that are being observed in some of the patients. In fact, they have been reported CD4 memory cells that have been 70 years old that they anchored these DVGs. And although the patients they seem do not have any traceable HIV detected, but they maintain some seropositivity exactly because of these DVGs. So it's an interesting observation, and there are a lot of scientists that have proposed DVGs for either using therapeutic areas as well. So it's definitely important to study them, and that means also that it hasn't stopped scientists from focusing on DVGs in this current epidemic and see how they might be playing a role in the evolution of SARS-CoV-2. There have been already a couple of studies out there. They looked at DVGs. It's mostly in the form of looking for bigger deletions, and you can see here the heat maps where they show the deletions and between and the jumps from one region to the other. And they have a nice heat map here showing practically what are the regions, the common regions for these breakpoints where we observe these deletions, because they reported mainly deletions for SARS-CoV-2. And this is work that has been done in mice, but they've also observed these deletions in human patients, as we see here on the right. The second figure from the other publication is reporting DVGs identified in human samples. So there are many viruses. Of course, there are not just these two, right? There's plenty of observational data that prove that there are DVGs in many RNA viruses, and they have a lot of different functions. This small collection of them, and you can see that there are not only a lot of viruses, but there are a lot of different functions that differ between each family or even within the same family of viruses. And in our case, at least, what triggered this tool and this publication, it was mumps virus. So we focused on mumps virus, and we created this DVG-profiler. Thanks to Dr. Bosma and Dr. Sauder from FDA, we had the chance to look at the data that they created with the NGS data and their wet lab results from a lot of the analysis that they performed previously, and they convinced themselves that there are DVGs in mumps virus, and we have to look at them. We have to see how they behave and how we can study them practically. So we came up with this workflow, this pipeline, which eventually led to the creation of this tool called DVG-profiler. The concept here is that we start from the HTS reads, or next generation sequencing reads, either that usually being that Illumina read for high-seq or MiSeq sequencing. And having these reads from mumps virus, the question is whether we can observe their junctions that justify these copy-back DVGs, or if we can find other junctions that can lead to potential DVGs. So we get these reads, and we firstly align them to the reference of the virus that we already have. And the whole point of the DVG-profiler is to get these alignments and process those alignments as an input now, and see and justify junctions through split reads being reads that originate from the same read, but that they align in different locations of the genome. And once we do that, try to find all the places that we report those hits and see how they pile up and report those locations. So this is practically the overview of the pipeline. Conceptually, I think it's an intuitive approach, and you will see later on that it pays off. And there were some similar applications as well. So just to give you like a more schematic description of what's going on, and that will help us to a little bit navigate the interface of HIVE and see how one can perform this workflow through the platform. The whole concept is that you have these reads, the short reads from one of the sequencing technologies, and you will align it to your reference genome, that being called the subject here. It could be one sequence, it can be multiple sequences. It doesn't really matter for the sake of the algorithm, and you will have the sequences aligned into multiple places. So in an ideal scenario where you have a read that it doesn't consist of any DVG, or it doesn't capture any junction, you have the entire read aligned somewhere in your subject. That would be great. That's perfect, and that is practically a well mapped read. But in the case of a junction or other events, you'll end up with some reads that will have a subsection aligning somewhere in the genome, in the reference sequence here, and then another subsection probably aligning in a reverse direction, and then maybe a third one overlapping or non-overlapping the other two somewhere else. And exactly how these subsections of the read, how they combine with each other, that's exactly the justification that we're looking for in order to assess whether a read is capturing a junction, right? A DVG in this case, and how they orient, let's say, themselves with regards to the read and the subject and the direction is practically what defines the potential DVG that this junction captures. So we have here like different cases. Here we see that these two, the blue and the gray reads, they're not overlapping, these split reads. They can also be partly overlapping, as we see here with the yellow and the gray, and they can be overlapping, but yet not fully capturing the read. As you can see, like this left subsection of the read here, it's not captured by these two alignments, or they can be overlapping or non-overlapping and capture the full length of the read. And in this case, this would be the preferential combination of the split reads, right? We want to focus on these ones and say that these are the ones that are capturing the junction. And now this is the whole idea behind it. It's rather simplistic, but it's what differentiates this tool between these and with other junction detection tools. The fact that it doesn't only detect deletions or insertions, but it can also detect 5' copy back DVGs or 3' copy back DVGs, which are the same type of 5', which are explained by the traditional replication, but also 3' to have an anti-genome, which can theoretically also be done. So in the case of deletion, you have the blue section of the short read, which is the leftmost section, let's call it, and then the first or the rightmost or last section of the read. If they are also aligning the genome in the same sequence, like the first being on the left side and the last being on the right side, and there's a gap here and you jump, you can see that this is practically what describes a deletion, and the distance between these two is exactly the gap, the deletion, the length of that deletion. But you can also observe that, from the reverse complement side, where both directions and what comes first of each split read is reversed. Similarly, if the second section is the last section is also second in the subject, but in reverse direction, this is the case that we're that describes a 3' copy back DVG. And if again the left side here is first, but the first side, sorry, here it's on the left side of the subject, and it has a reverse direction, then this is the definition of a 5' copy back. And finally, when both sub-reads they are in reverse direction, they are describing an insertion, which is a little bit more intuitive if you look at it from the reverse complement side. This is a case of an insertion where you have the polymerase here going forward, then at some point jumping back and in the previous point and continuing. Practically, duplicating this this region. Excuse me. So, so as I was trying to explain here, these are the different cases, but as you can see, in each different case, there's a region that it's overlapping. Like there is this overlapping region here between the subsections, and where you draw the line, where you jump here and you go there, that's not a straightforward call. That's something that we have to look into, right? Whether we should jump and call the junction point on this end or this end. This is an easy call. We just look into the overlapping region and we say that if we start here, then we include some mutations there, or if we go all the way to the end of this the blue side alignment, then we include some mutations on that end. So that's not the way to optimize the score. So that was the goal: how to create the right junction or detect the right junction, so it optimizes the score of the combined two split reads, the score of the alignment, that is. And you can see that this is possible not only on one direction, right? You might have two reads actually being a better combined alignment if you include if you conceive them as a separate as a reverse direction, and that in that case that would create a much better score. So these are all like features that make this junction detection approach unique compared to other techniques that are out there. And the reason is that you need these techniques, and the reason is simply because we're dealing with viruses, and these viruses, they have a lot of mutations. These mutations are there just because the virus evolves. We cannot disregard them. We cannot remove these reads, as a lot of splice junction algorithms do for RNA-seq, for example. These are exactly the reads that justify these DVGs, and that was the need for this tool, to look into it in such an sensitive way, including all these mutations. So we did that, and the reason we even started looking into this tool is because, as you can see here, the NGS reads that were produced by for three different viruses, mumps viruses, that is again, and then later aligned into against the reference. And here we can see the pileups, the profile, the positional profile of these alignments. We can see that there's an increased depth of coverage in the right-hand side, like the 3' side, 5' side, sorry. So there was already an indication that there are DVGs here, and these were the data sets that we looked into finding these DVGs. And our collaborators, Dr. Sauder, and Dr. Postman, they looked into PCR approaches where they created different sets of primers that capture these DVGs practically, and they were able to observe them, and it was a matter of whether the tool now can actually return these the same results and even more, and it did. I don't think that there's really a point of like just going through the results here and proving that we did detect these DVGs, but the tool has detected those and even more. And I'll come back to that in a later slide just to talk a little bit more about the nature of these DVGs and the evolution of them. So after we run that, we just wanted to see that the tool is actually performing as it's supposed to, and that was just a proof of concept, trying to minimize the length of the read that we allow during the alignment, the minimum split read, the length of the minimum split read. And you can see here that the smaller the split read, the greater the sensitivity. That makes sense. It's able to capture even more reads around the junction, and that later justifies more junctions. That increases the sensitivity. And of course, we go down to 10 positions. Otherwise, that explodes the alignments, the number of alignments. And interestingly enough, we also run some reproducibility tests here. And Dr. Sauter and the lab, they performed 2 different sequencing runs, and they observed how the tool can consistently identify these junctions. And here they're grouped into 2 different sets, the copybacks and the indels. And as you can see, the copybacks, they're very strongly reproducible between these 2 sets, and indels, they are as well reproducible. As you can see here, the R regression test is not as high as the copyback, but yet these are encouraging results. Interestingly, there was another tool that came out when we were working on DVG-profiler, DI detector. I don't think that's really worth getting into the technical details here. I just want to bring it up because I found it an interesting approach and highlights the difference between approaches within HIVE and approaches outside of a unified environment. I'm guessing the reason was because the available tools to that group was not the flexibility of having their own aligner. What they try to do, instead of examining all these different alignments of a single read, what they try to do is try to create multiple sets of one read by taking one single read and splitting it into multiple positions, practically creating all these potential alignments that we capture with that one alignment step, which of course creates a much greater initial data set to work with and slows down the process. And that's of course also justified by the results. We just run a comparison here. I don't think that we need to focus on the accuracy. They were very similar in accuracy. But it was also interesting that you can see how the other tool explodes in time with the size of the data set, and how in comparison DVG-profiler, it just practically stays stable compared to it's linear or rather sublinear, but in comparison to an exponential growth, it looks like a flat line. So that was an encouraging also step that we found this publication, which is doing something very similar. We were just more flexible, and because we were using our own aligner, which is optimized for viruses, we were able to actually achieve higher sensitivity. So, and this is how you can do it here. I can just go a little bit through the slides to show you the environment. This is what you will see when you log in to HIVE. Of course, you won't have all the data here that I have in my account, but you will have your HIVE space, and you can upload your own data. There's a way to click and navigate into different pages where you will be able to upload your own data sets, your own NGS reads, your own references, your own databases. You can do all these things, and then you can also run the tools that you will find available through HIVE. And in this case, I'm just showing you these 2 data sets, these 2 reads, one per then, actually a set of reads for one of the viruses that we processed, one of the mumps viruses that we processed. And you can see that when you upload these data, this type of data set into HIVE, we already run some analytics where you can see the summary here of the base counts for these reads and some other information. And having chosen these 2 reads, you can jump and say that I want to align those reads, which is the next step of the workflow that I showed originally. And that will take you to the hexagon aligner page. It's a specific aligner developed for HIVE, and it's highly optimized for viruses, so that was a great fit for this workflow. And you will see a set of parameters. This is a simple set, the general exposed parameters. There are more advanced parameters if you want to play with the algorithm, and you can read more about how you can do so and all these different features. in the tutorial here on the right, but the whole point here is that once you jump to this page from the home page, you'll see your reads selected already here in the input section, and then you will have to choose your reference genome that you want to align those reads against. This case this was the name for the mumps virus reference that was selected, and we want to keep the most of the things here. The default. This is the minimum. Oh, sorry. This is the minimum length that you want to allow this split read to be, and it was chosen to be 17 base pairs. In this case, can make it even smaller or even less. And then the key point here is that you have to say that I want to keep all the matches within the acceptable threshold, and that's what practically allows the next step, the DVG-profiler, to do the magic, right? To create all these combinations of these mappings of the read. So we keep all the matches here, and then we perform the alignment. And you can see in the alignment we have a lot of hits here. Some of them they didn't get aligned for a number of reasons. You can navigate through the computation of the aligner. Just look at the alignments themselves, and download the results if you want, or you can just simply choose the section what's next and jump to the next step of the pipeline, which in this case is the DVG profiling. And DVG profiling, once you do that, it will bring you to the page, the relevant page here, where it will populate already the alignment as an input of the tool, and it will let you alter the options here, the different parameters according to the algorithm as we described it, right? Some of them being the maximum distance between these split reads, as I'm showing here, or the maximum overlap between two split reads. You can choose to filter some of these combinations based on these parameters. Also, there are some more intuitive parameters here, like the depth of coverage. The depth of coverage that justify one junction, everything that is below two reads supporting this junction, it will be filtered out. Or the minimum length of the aligned read. Practically here, it's a combination. Or it's the total number. It's the total length minus the distance that we might have here. And there are also some more advanced features that, such as the overlap score threshold. Practically, if you have an overlap, as it was described before, as it's shown here, in order to make sure that that we say that despite the fact that there's an overlap here, we're gonna keep the read if the overlap creates a combined alignment score better than the non-overlapping combination, another possible combination. And that's the percentage practically here that allows you to filter based on the mapping score. And there is also the mapping against the subject, how much of the subject you want to align to, because you don't want to have an alignment against the same genome twice, for example, if you're not interested in snapback DVGs. And the grouping, which is required for a later stage, because due to the evolution of even DVGs, you might not get accurate, stable junction calls, even though we're trying to call these positions in a stable way. And also, some cases you might have many, more than 2 or 3 split reads, or even more, which they create more than one combination. And in this case, you have to say which combination you want to choose. Do you want to choose the one that creates the best alignment score, or the longest read that's been mapped? And this is just for reporting purposes, whether your genome is given as an antisense so we report the DVGs or the 3' or 5' DVGs. And here you can see practically the output. After you run the computation, you'll see the positions that we detected. This is on the left side, and the straightness as described in these 4 cases, and then position on the right, and then the straightness again. And for simplicity, instead of just looking at the straightness and trying to figure out what it is every time, you can just look at it here and see this is a 5' copy back, This is the length that's been created in this case, the delta between these two positions, and the number of hits that's supporting the position. And you can see that we report both the forward and the reverse hits as they've been described in the previous chart for the reverse complement junctions. And you can see that there are some cases like this one here, where we do report a lot of hits in this position, but there are neighboring positions that are reporting some of the hits. So and the reason is simply because DVG is also a viral genome, but that although it's not able to replicate itself, it sometimes it's getting replicated through machinery. The rest of the virus is present in the cell, and that means that it does evolve on its own. And we've seen that in the data set here as well. So you can see that if we plot the data here, we see regions that you look closely, there are similar regions that one big event is reported, and then there are some small hits left and right that are reporting the same event. And in order to consolidate these results, so we just run a 2D peak detection algorithm. It's like a 4-way connectivity, not an 8-way connectivity, where it will just collapse all these hits into the peak. And that was just to give you an idea of what's the second tab here of the results, where it reports the same junctions practically, but they're grouped through this process that has already been run for you. And it also includes information about these groups, where was the first one reported and where was the last reported. And here is just the position of the peak as reported. And that also leads to a smaller set and consolidated set of junctions. So, and the reason, as I said, that we do this grouping is because some of these DVGs that we observe, they're not 100% identical, either through noise or through some evolution. They do accumulate some mutations, and these mutations, they tend to move these reads, the alignments of these split reads by 1 or 2 positions. And not only that, like we also observed here, as I'm showing from the publication, some species of DVGs where they not only have copy-back DVGs, but they also included additional deletions, and this was observed through different loop sizes. And I'm just highlighting 2 of those. So there's a clear evidence that these DVGs, they do evolve. We do observe some variations there, and it's as if they are part of quasispecies themselves. And I just want to touch base a little bit on the quasispecies here. I think I need to kind of like move faster on these slides, but quasispecies in general, it's a mathematical definition in the space of RNA viruses. It's also attributed to the low fidelity RNA polymerase, but nonetheless, it has been firstly defined as a mathematical model. And despite some debate in the field on whether we should call them quasispecies, I think everybody does that right now, or a lot of people are doing it. So I'll continue doing that, giving that heads up. The whole point, though, is that this quasispecies just describes this cloud, this swarm of variants, and the need to resolve all these variants from one sample. There's even a formal mathematical definition of the problem. It's called quasispecies spectrum reconstruction, and it's what we see here. Given a set of NGS reads, how can we resolve back these quasispecies and not only to resolve their consensus sequences, but also their frequency? And this is actually the problem that we studied as well, and we've published an algorithm also available in Hive. I don't really want to go into the details of the algorithm. I just want to show you here that conceptually an algorithm that starts with your NGS reads, you align them to your reference, and then it's an approach that it starts with a window, it accumulates those sequences, and the window examines these sequences of this particular region and tries to identify the mutations and split them into different windows where each mutation is creating a single consensus here. And then these windows now, these multiple windows, they all like move and slide to the next step. And the second part of the window is used, this is a little bit off, is used as template for the next step. And that practically creates a stepwise approach for this algorithm. So it's a sliding window. And you'll see where I'm going with this and how this is linked to the DVG. So the whole point of what I wanted to show you here is that this sliding window, it creates eventually these graphs, which are also presented as a graph genomes in the world of sequencing assembly, the human sequencing assembly. That's a relatively recent development, but they're also here in virology a way to describe quasispecies. It's slightly different from the Sankey diagrams, but they're similar. You can see the width here represents the depth of the coverage at each given position of the strain of the quasispecies population that we described, and the bifurcation, small branches that come out and then back into the original clone. And this is practically a way of reading the different species the quasispecies that we observe in the sample. Here you can see that this is a potential path. There's another path. This is another path as well. And this is not, because the middle part here is generated from the lower, and the rightmost part here is generated from the upper. So this is not really a way to follow this trajectory. So just wanted to show you how to read a little bit these graphs. All these are available through Hive. We've done the same thing with mumps, and we are able to resolve these different quasispecies. And this tool has been available through Hive. It's called Hexa Hedron, and you can download it and work with your viral data set. But and you can do so the same way. You can upload your data to Hive as shown here. And you can jump to the MAFTA aligner, and you want to do that because the most important things in these tools is trying to bring everything into one common coordinate space. So you have multiple references that you want to align against, then you want to make them all report in the same coordinate system. So you run the multiple alignment sequencing to do so. And after you've done that, you can jump to process your reads now, and you can process the reads that are showed originally and choose the reference set. As I said here, like we would use multiple sequences. This is the name of it. This is Hive showing you that this object actually consists of 3 different sequences. And in this case, there are 3 different sequences from the Sabin family. And they are, and then run the alignment again, and this time without specifying all the matches, right? And after you run the alignment, then again you get to see the results of the alignment here, and jump as a next step to the Hexa Hedron alignment. Here there are some of the input parameters you can choose. I don't want to focus on how to parameterize this tool, but the fact that you can see that in this case that we were able to resolve these 3 quasispecies. But as I mentioned, that in this case, in this example so far, we see only quasispecies that are from one end to another, and the whole goal is to try to create quasispecies that are, or rather, report sequences that are fully resolved, that's called global sequences. And there has been work done also in collaboration with Dr. Mazunder's lab and Dr. Feng's lab from Duke University. And they've applied this tool on HIV virus, and they were able to use it for subtyping, and they were able to devise this pipeline where it just starts from the reads. It picks the best set for references for this type of analysis, and then all the way resolve the quasispecies, and it does it so in a global way. So in this case, it knows how to jump, how many times it should jump from condic 2, for example, to 3 and 4, and not 5, or how many there might be from nature of 2, 3, and 5 in this case. There are ways to do it. I don't want to focus on the computational side. We can talk more if you want, and if you have any questions. Just want to show you here that the tool at the end it allows you to not only display the the Quasispecies as this graph that I was showing previously, but it allows you to export the sequences as shown here. You can choose which clone you want to focus on and then display it here and even download it. You can print it consensus, you can print the frequency of each one, you can resolve it either the condigs or just infer the global sequences based on the Monte Carlo algorithm that has been described before. And this work has been done and has been actually tested against multiple approaches, one being the Genius assembler, proprietary de novo assembler, practically, which cannot really resolve the Quasispecies, but it can identify all these positions that are arbitrary positions, and it reports those. And you can see that it reports them accurately enough, or at least it was in accordance to HIVE, Hexa Hedron, resolving these different condigs. And this has been further proved that they exist through single genome sequencing here from work from Dr. Feng's group. And you can find out more about this process from the publication here. The whole point, though, is that we've studied significantly Quasispecies, and we've also studied significantly DVGs, but we haven't really focused on a combined problem, right? The key point here that and something that hasn't been really communicated often in the world of Quasispecies spectrum reconstruction algorithms is the challenge of big insertions and deletions, let alone copy back DVGs that have even more complicated coordinate systems. It's exactly that. It's the fact that big insertions or deletions, they create a big problem because they mess up with the coordinate system that it needs to be there for the resolution of the Quasispecies spectrum. In fact, there are tools that will disregard completely your deletions detected in your NGS reads. The NGS reads, they might have them, but the tools will completely disregard them because it's a hard problem. So, and I don't think that's a good approach. And frankly, Hexa Hedron is trying its best to capture both deletions and insertions that have been observed in the samples. And during the whole process and these publications, we observed that there are also potential particles there that they do not easily fit in the mutual coordinate system that we try to describe. And this can be very much the reason, and DVGs can be very much the reason for this observation. So maybe what we need to do is we need to redefine the definition of the Quasispecies spectrum reconstruction. We shouldn't be looking into resolving these sequences that simply have single point mutations or short deletions or insertions, but we should include DVGs that might be present in the sample. So we should be looking to identify those DVGs along with other consensuses and quantifying and their abundance. I think that that would be the best way also to capture DVGs and remove some noise, or some perceived noise rather, from Quasispecies. So I see these two challenges, two mathematical challenges, as one in this field. And I know this is mostly wishful thinking right now, or like future work, because we haven't worked on that, or don't have anything to present to you right now. But I think that's the way to move forward. Take the same pipeline that has been used has been developed for Quasispecies, and improve it, and try to integrate the concept of DVGs, and try to include them in the references, and try to resolve both DVGs and Quasispecies as defined with the previous way. And I hope to be able to continue working on that and improve not only the pipeline but the tools, and possibly introduce new tools. As you can imagine, that we still need a way of getting the actual sequences out of the DVG profiler, and this is something that we will be working on. And with that, I wanted to thank a lot of people from our team, which they contribute significantly in a lot of aspects of this work and keeping Hive platform and providing all the tools that are required to do this kind of work. Of course, George Washington University and Dr. Mazunder's lab, they've used the tools and they created pipelines and they created a lot of features that made this work possible. Of course, also our collaborators from OVRR and Dr. Sauder and Dr. Bosma, they've done great work for DVGs, and Dr. Chumakov and Dr. Lasry. They've helped tremendously with Quasispecies work, and they produce a lot of the samples that I described today. Of course, from Duke's lab, from Dr. Pankau and Dr. Bachnauer, that they helped a lot with the Quasispecies implementation on HIV. And I think with that, I will stop here and ask if you have any questions.
All right, thank you very much. So there are two options. You can ask questions directly. I have allowed you to unmute yourself and ask questions, or you can also type in your questions here on the Easy Retro page that we were showing. I see a question. To use sophisticated tools like this in regulated applications, can you comment on the kinds of in silico and then also biological samples that might be needed to optimize and qualify a pipeline for a particular use?
This is an excellent question. This is a question for most of the tools that we should be creating. Both of the tools have been thoroughly examined with in silico and biological samples, to be honest. And the DVG-profiler has been tested against a lot of different sets, in silico sets, and to detect the limits of different parameters and the behavior of the algorithm when you change some of these parameters. Of course, you understand that this is something that you can do depending on a certain scale, depending on the complexity of each algorithm and the number of different options each algorithm provides. In this case, given the rather small number of parameters, we were able to examine the behavior of the algorithm by changing most of them, I think all of them. And we were able to detect the different, limits of sensitivity and accuracy when we change for example, the length of the overlap or the minimum length of the split read, and the alignment score of the overlapping regions. We were able to do something similar with the Quasispecies, the Hexa Hedron, the Quasispecies spectrum reconstruction algorithm, and that was the main focus with the in silico dataset. But this is the in silico dataset. Once you move to biological datasets, it's only so many you can generate to test the performance of the algorithm. I think the main focus for biological samples is when you're trying to assess their reproducibility or trying to detect the noise that's being captured or can interfere with the performance of the algorithm. And that's something that at least the Quasispecies the Quasispecies spectrum reconstruction tool, Hexa Hedron, has been tested thoroughly by the implementation of the HIV samples, and the integration to the pipeline that was processing the HIV samples, and the DVG-profiler was also examined against multiple biological samples for reproducibility. But yeah, this is an ongoing challenge, and specifically for even more sophisticated tools, this is something that's hard to assess with one single publication. And if you go into the world of aligners, for example, this is becoming increasingly more complicated. And that's why the community in the field just slowly focuses from the use of one aligner to another, and it's through experience and repetitive work that we develop some confidence on which tool is better implemented or which tool is better suited for which samples and which type of analysis.
So I'm going to add another comment here. So this is Raja, and what we do believe in is that figuring out if the tool is working the way it should, and are we finding what we are intending to find in the first place or hope to find in the first place, is going to be an ongoing effort. But what can be done right now is recording all of that information in such a way that people can reproduce it, replicate it, and so on. So even though today's talk is not about biocompute, there's another project our group and Dr. Karagiannis' group and many others at FDA are involved in, is the biocompute, which is what was done in silico. And then you then use it to better understand using wet lab procedures. That's what we did with the Duke Vaccine Institute for the Hexa Hedron, on trying to see what we were trying to see in silico, can we actually replicate it in the wet lab? And designing the experiments is also really, really important, both in silico and in the wet lab. And we are seeing this again and again and again. So taking tools from off the shelf and trying to use it is step one. But then when people are really trying to understand what is going on in a single specimen, NGS is really powerful only if it is used in, its full capacity. That's what we feel. There is another question. Let me see. Going to the listed URL for Hive Hexa Hedron for installation instruction at the bottom, it sends you to the Hive page, but the page is sent you. Okay, so if you want to install Hive on your own server, there is a GitHub installation that you can download, but it does not have any support. We do not have any funding to support provide technical support for Hive installation. On the other hand, what everybody on the call can do, my group has some NSF funding to provide Hive usage to people. So you can sign up for the Hive usage, and we will create an account for you, and you can try out DVG or Hexa Hedron or Hexagon. And it will be free. It will be free. You do not have to pay anything. There is a cost to download data, but it's very, very minimal. So because NSF doesn't cover that, or the way we set it up, it doesn't cover that. It's on AWS and it's not there. So if you are not an FDA employee, that's one option. If you're an FDA employee, actually, Dinos can tell you how to use FDA Hive. You want to say a few words on that?
Yeah, thank you, Raja. Well, if you're an FDA employee, I think it's a pretty straightforward approach. You can go to hive.fda.gov, and you'll find links and information there that you need to register. You're practically sending us an email to activate your account once you register. And that's pretty much it. We might have to, check your credentials and make sure that your supervisor is up to date with the registration. And depending on the projects that you might have to run, we might chat a little bit to see the type of usage that you want to have on Hive. But given the fact that we have both regulatory instance and scientific instance, then we can, I think we can easily accommodate your needs. But registering to Hive, that should be the easy and first step that you can do. I will also try to post the link here and some answer to this question if it's possible. And I hope that will answer all of your related questions as to how to register into Hive.
Yes, and we are always looking for collaborations. There's another question that came in. Did you get a chance to run the DVG pipeline on the HIV data that Duke generated and find any DVGs? I don't remember you doing that, so unless you did it.
I can shortly answer that I haven't done that. And DVGs and HIV, it's slightly more complicated, I think, due to the way that the replication cycle of HIV works, and it might require slightly different references. But that's something that we could possibly do. And essentially, just based on what I've observed in the past, the longer deletions, I think that it would be definitely be reporting some deletion type DVGs, but I haven't run this tool yet on this data set.
More questions? Anybody who wants to use the tool, get more feedback, or any other viruses? For example, has anybody looked at your end on the coronaviruses, or it is not something that you think there is enough evidence so far?
So the work on coronavirus is something that I recently discovered myself, that there are some people that are working on DVGs with coronavirus. It's something that we can look into. We have a lot of sequences from NCBI into HIVE and HIVE. We maintain a version of NCBI's database for SARS-CoV-2 sequences, and we could be looking into these data sets, but these are not NGS reads. So I'm not sure, if we could possibly capture DVGs from the reads themselves. We do have some collaborations that we might be able to generate. Well, we might be able to detect these junctions from these reads, but in terms of patient collected samples, we haven't had a chance to work with this kind of data yet.
More questions? Actually, I'm going to put the questions up. I'm going to, or I can read the questions to you.
I can
or you can read them also, right? Please go ahead.
Yeah, one says here, let me see. Okay. Okay, so there is a means of identifying and quantifying DVGs and maybe even Quasispecies. Is there any sort of scientific working group that addresses how to interpret these? Also, assuming we also wanted SNV analysis, is that by Hexagon? Yes. Well, that's a good question. I'm not sure whether there's a working group to address the biological challenges of how to interpret those genomic species, but it's a great suggestion. I believe we could be focusing on how to interpret these. This focus and this presentation and these tools mainly focus on the analytical side and providing the data to interpret them later on. And I think that's a pretty unique approach so far. I haven't seen anybody out there speaking of a combination of DVGs and Quasispecies. And the other point on whether the SNV analysis can be done on by Hexagon, that's absolutely true. Hexagon will return you the frequencies of the point mutations or essentially small insertions and deletions per position, and you can focus on CNV through this analysis with Hexagon, which you can perform after you remove, for example, reads that have been attributed to DVGs. Which brings me to the next question, actually. The next question is, would identification of DVGs effectively lower the percentage of SNVs? Well, I guess that's the hypothesis, right? That's what we're claiming, that some of these mutations, they can be coming from falsely mapped reads due to DVGs. And if that's the case, then yes, DVGs, and knowing the junctions and recreating these sequences and providing them in your alignment step as a decoy, let's say, as a sequence that will draw these reads to the DVGs rather than the reference genome, will essentially reduce the percent of SNVs, either the sheer amount of them or just the frequencies reported.
All right, thank you very much. And if there are more questions, please let us know. And with that, I think I'm going to close the meeting for today. Really appreciate you all taking the time to join us. Hopefully you'll be able to join us when we have our next seminar/Hive talk. Bye, everybody. Thank you, Dr. Karagiannis, and thank you, everybody.
Thanks very much. Bye, everybody.